So last time we talked about how to cut DNA and place them into the plasmid, and then we put the plasmid into the bacteria and grow it. But one application, one example on maths, nobody will do this, not the math scientists will do this, but something to think about, something to ponder over. It is this one. Look at this diagram. This is something I've got. You don't have it in your PowerPoint, but I will put it up for you guys if you want to take notes and write a couple of definitions that the application of this method. You know, ampicillin and kanamycin. Can you guys see that from back there a little bit? Yeah. Ampicillin and kanamycin, these are the two drugs that kill bacteria. They are antibiotics. Okay, so this bacteria has an BNH1 and HIND3. They are restriction enzymes. They are name of restriction enzymes. Restriction enzymes, it means enzymes that can cut the plasmids. And then T4 ligase is the name of the ligation. It puts the DNA fragments together. Okay, these are the things you already know. Okay, now you have a bacteria that has this plasmid that has that gene called AMP. That gene is making that bacteria resistant to ampicillin. It means if you pour ampicillin as a liquid, you pour ampicillin on top of that bacteria will not die. Just like the plates you have. You all follow what I'm talking about. So, you have some bacteria here in this plate that you dump ampicillin on top of it, they will not die. But if you dump kanamycin, am I making sense? The gene, this kanamycin, if I dump kanamycin on top of it, it will die. They all die. Am I making some sense? This one, this bacteria, these bacteria, if I dump ampicillin on top of them, they will die. If I dump kanamycin, they will not die. Do you all follow what I'm talking about? So this one, if I have a big marker, this one I should write down on K on it. Ampicillin, AMP, and this one I'm going to write down KAN. KAN. You guys see that? AMP, KAN. If I dump ampicillin on this one, they will not die. I dump kanamycin, it will die. When I say AMP, it means they're resistant to it. When I say KAN, it means they're resistant to kanamycin, but ampicillin will kill these guys. Am I making some sense? Now, I want to make a bacteria. They do this daily in the lab every day. I want to make a bacteria like these that are resistant to both ampicillin and kanamycin. This one, I'm going to write it down, AMP plus KAN. So this bacteria here, they're all resistant to both of them, resistant to ampicillin and kanamycin. That's not a good thing to do, but just for science purposes. Stephen, am I making some sense? For science purposes. So you are going to make a plasmid that has both ampicillin and kanamycin. Do I make sense? How you do that? Something you've done on last time, whenever it was, Wednesday. Here we go. Something you've done on Wednesday. So how you go about it? What you do, first of all, you put this one, the AMP you put it through restriction enzymes, you introduce, just like, you remember at the beginning, the very first thing I did to your vials, the suspect 1, suspect 2, suspect 3, crime of scene, I added restriction enzymes. That's what you do. You add restriction enzyme to that plasmid, and then the plasmid breaks, you have sticky ends. You remember sticky ends? You have sticky ends, and then you introduce to the plasmid of the kanamycin, the bacteria with the kanamycin resistant, and they break. Okay? So these two, now you have fragments. Just like suspect 1, suspect 2, suspect 3, suspect 4, suspect 5, and from scene of the crime. So you do have fragments now. Then you do gel electrophoresis. When you do gel electrophoresis, what will happen, you have bands, you know your restriction enzymes worked. That's the purpose of gel electrophoresis. You run through gel electrophoresis to see restriction enzyme worked or not. In this case, it did work. Okay? Then you take ligase, T4 ligase, and you put these two genes, sticky ends, you put them together, and now you have a plasmid with both ampicillin and kanamycin resistant, and you put those plasmids, you put these plasmids into the bacteria, and you grow the bacteria, and you have a plate. You have bacteria now. If I dump ampicillin and kanamycin on this one, neither one of them will kill this bacteria. That's one of the, and then if you were wondering how do they make, for example, how do they make cotton plants that are resistant to pesticides? This is it. They take a gene from a cotton plant that is resistant to pesticides, and they put it together with the gene of the plant, the cotton plant that is not so resistant, and then they grow them and they become resistant to cotton. Okay, this is the technique that you remember. If we go back, I'm going to flash back quickly here in a minute. Oh, you remember this one, the bacteria. They have two application, one application to make growth hormone, a person grows tall, or insulin. I gave you the example of insulin. And the other application, you take the plasmid, you take a gene and make a resistant plant, or make bacteria that eat oil. Okay, that's just the one that I talked about right now applies this one. Oh, these are PCR. We talked about PCR. That's the one that applies to the other aspect of it. This is not an experiment for making insulin or glucagon, or making insulin or growth hormone. Does anybody have any question? I know you had a rough weekend. It was Labor Day, a lot of celebration for Labor Day. Right? Yesterday was Labor Day, right? You didn't know that, huh? I said it. These guys came late. Can I repeat my story again one more time? Ariel, she's half asleep, half awake. Yeah, I mean, you can board us some more. It's okay. Stephen, did you know that? You didn't know that.
What?
That Labor Day was yesterday.
It was?
Yes. A lot of people don't know this. The rest of the world celebrate Labor Day on May 1st. That's it. Labor Day in the entire world. United States in 1930s, the Congress said, no, we do not want it May 1st. Because that would be too much in the shoes of communism. You know, communist countries, their base is based on labor. So they said, no, we don't want that. Let's move it to first Monday of September. Whatever that is. That's how Labor Day was. If you don't believe me, look at your resources. Google. You guys don't even know where library is. Yeah. Google, YouTube. Look at it. Yesterday was Labor Day. whole world, everywhere in the world is closed. Most places, factories and other places close May 1st. As much as they can. As much as they can. Alicia, you know now. What is the May 1st, Labor Day, May 1st. But anyway, let's go. Gene cloning a gene. I think I talked about this. Everything we talked about it.
We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid. The ampicillin in the medium limits growth only to plasmid-containing cells. But which cells have plasmids with the human gene? Bacteria without human genes have intact LAC-Z genes. This allows them to break down a substance in the medium that stains their colonies blue. Thus, the bacteria with human genes are in the colorless colonies.
Yeah, that's another concept. I didn't talk about it because we don't do much of it in the lab, so I'm not worried about it. Cloned organisms and stem cells are useful for basic research and other applications. Organismal cloning produces one or more organisms genetically identical to the parent that donate a single cell, and I will talk about this. Stem cell is relatively specialized cell that can reproduce itself indefinitely, and under certain conditions can differentiate into one or more type of specialized cell. More wordy. Where is the? Here we go. Great. Okay, I guess those are prelude. Reproductive cloning of mammals. I don't know. You guys have heard about in 1993. For the first time, they cloned a sheep, a mammal, a vertebrate mammal. That was the first time, and now we'll talk about it today a little bit. Something you should know about it. Okay, in 1997, I'm sorry, I said 1993. In 1997, Scottish researchers announced the birth of Dolly the sheep, a lamb clone from adult sheep by nuclear transplantation from a differentiated mammary cells. And I will talk about that a little bit, where the mammary cells came from. Dolly premature death in 2003, so about 5 years later, as well as her arthritis led to a speculation that her cells were not as healthy as those of the normal sheep, possibly reflecting incomplete reprogramming of their original transplanted nucleus. When they cloned, that's what one of the problems with cloning is. You know, you've heard about cloning animals. They clone animals nowadays. And you take your dog or your cat and you want it cloned, they will clone it. They will clone your dog or cat. It is possible. They do that, the exact same duplicate. However, there are a few problems with it. One problem is your dog or cat that get cloned might not act, they do not behave same way as the dog or your cat you have. That's number one. Number two, they usually die earlier. Okay? They die earlier because you're using an older chromosomes. At the end, you remember, and we talked about this, at the end of chromosomes there are telomeres. These telomeres get cut off after every time the cell multiplies a little bit of telomere get cut off. Okay, that's what we put these chromosomes in these animals, and it's a chromosome on older animals. That's why these animals have arthritis. They're older animals. The chromosomes are older animals, but the animal was just born. Okay, so that's the problems we have. A few of the problems we have with Crohn. Okay, what happened in 1997? What happened? And these people, I don't know, they have the Nobel Prize for it or not. They will. I think they will. The Scottish scientists, if they have not won it, they will. What happened? They took a sheep, white-headed sheep, that the mammary cells. How many chromosomes are in the mammary cells of white sheep? Let's say sheep has. I gave you these numbers at the beginning of semester somewhere along the line. You didn't have to memorize it. I don't have to memorize it. But let's say that sheep, the mammary cells from sheep has 46 chromosomes, same as us. Let's assume that. Every species has a different number of chromosomes. Some of them have the same number. Has 46. And then they took the egg from a black-headed sheep. You see the black-headed sheep, egg cell donor, and of course they removed the nucleus from black-headed sheep egg. They had to remove the nucleus. And then they took the nucleus from mammary cells of a white-headed sheep and put it in the egg of black-headed sheep. Do I make sense so far? And then, of course, they pass electricity, they give it electric shock, because that's what they say, anytime sperm enters the egg, it's a theory, it's an assumption, I don't know. In the fallopian tube of female, there is an electricity, there is a shock. The egg has to wake up as soon as the sperm enters the egg. So what they've done, they passed an electric shock there. It let it grow. It became blastula, something you're familiar with, mitosis. And they grow it in a dish. They grow the fertilized egg in a dish like this, and then eventually they took that morula, or blastula in this case, they put it in the uterus of this sheep, whatever sheep that is, the black-headed sheep, doesn't matter. And then when the baby born, the lamb was born, it was clone of that sheep. Do I make sense? It had to be clone of that sheep. It had nothing to do with this sheep, except one thing. What is it? What is it that had to do with this sheep? What is it? This is the exact clone of that sheep, the white-headed sheep, but it had, you should know, based on the information I gave you in this class, this sheep had something from this black-headed sheep. And what would that be?
The mitochondria.
Very good. Somebody is with me. The mitochondria. When they removed the nucleus, remember they removed the nucleus from the egg of that sheep, they still cannot remove the mitochondria. So this animal is not 100% clone of that animal. It's about 95, 96, 97%. What is the other 2-3%? Mitochondria. Very good. Mitochondria. You cannot, and mitochondria always comes from mother. Mitochondria always comes from mother. Okay? So that's how they clone the sheep. And does anybody know why they call it Dolly? Why not Amir? Amir, I think that's discrimination. I should sue them. Why not Amir? Why not Stephen? Huh? Stephen. Why not Stephen? Why Dolly? Huh? Dolly was the name of a country singer, Dolly Parton. You all know Dolly Parton? She's a country singer. Not James Bay, sorry. Not James Bay, but Dolly Parton. She's a country singer. And she has big breasts, I guess. That's what it is.
She has big hair too.
A big hair too. And when they removed the mammary cells, the Scottish scientists, they said, what shall we call that sheep? They called it, because the cells came from mammary cells. They could grab any cells from any body, any part of the body. They could grab the nucleus of bone cells, muscle cells, skin cells, But in this case, they pick the cells from mammary cells. Okay, so they call the sheep dog. Something for you guys to do. But anyhow, here we go. I guess they again in a larger version of it. Okay. Since 1997, cloning has been demonstrated in many mammals, including mice, cats, cows, horses, mules, pigs, and dogs, and many more. In this way, they are doing cloning animals that are going on extinction. You know, it is possible animals that they're becoming extinct. They are cloning them, but again, they do not live for a long time. Carbon copy CC for the first cat clone. However, CC differed somewhat from her female parents. Cloned animals do not always look or behave. Already talked about that. Look or behave exactly as their parents. Here we go, the clone cat. Stem cells of animals. Stem cells are relatively unspecialized cells that can both reproduce indefinitely and under certain conditions differentiates into one or more specialized cells. Here they are. What they do, I don't know, you remember history of President Bush. President Bush banned cloning of embryos. He said if you want to use for research purposes, for federal grants, if you want to use clone embryo, you cannot do that. But what happened, he suggested that we should use stem cell, the stem cell that are adult stem cells. We have two types of stem cells. One of them is, well, we have more than two, but two that I'm hoping you would know. One of them is called embryonic stem cell. And the other one is called adult stem cell. Adult stem cells comes from you and I. Okay, they take a cell from your skin, easy to get, and then they differentiate it to your liver cells or your brain cells or your muscle cells. So skin cells can differentiate to liver cells, to neurons, your nerve cells, to your cardiac cells, your heart. Okay, that's what they're showing you. That's called adult stem cell. What is embryonic stem cells? What happens in the United States in fertility clinics? They collect sperm from the husband and egg from mother. Okay, they fertilize them under microscope in a dish, and they grow it to this blastula stage, and then they put this blastula inside of the mother's uterus. This is for those parents, those people who cannot have children. They usually do that. They take the sperm, they take the egg, they put them together in a dish, and then after mitosis occurs, you know, fertilization and mitosis, 2 cells, 4 cells, we had model here for you guys to look at. Then you have this is called a blastocyst. Then they put the blastocyst inside of female's uterus, and the baby will grow. 9 months later, the baby is born. You heard about it, probably, possibly. They call this test tube baby. Okay, now, what happens, a lot of times they make these blastocysts. And these after the husband and wife, the couples have their own children, what they made a lot of these. They don't make only 1 or 2, they make a lot of them. If this month doesn't work, the blastocyst does not implement itself, they call it implementation. If it does not implement itself, they go next month, they give them another one, until the women accept that and then become pregnant. However, at the end, they have to flush these down the toilet. A lot of these are available in laboratories. They flush them down the toilet. what will happen? You know, the research said, okay, the people who are doing research, they said, do not throw it away, please. Give it to us, to the researchers, and they take that cell and they make it brain cells out of it. They take that cell and they make liver cells out of it. Same thing here. But they take it from an embryonic stem cell. What is the advantage of embryonic stem cell over adult stem cell? Embryonic stem cell is easier, Ariel, is easier to convert these cells. They are not differentiated yet, right? It's easier to turn them into brain cells, liver cells, muscle cells, any other cells they want.
You could grow
That's
into
easy.
anything, right?
Huh?
You can make them into anything.
Anything, any kind of cells you want. Of course, for adult stem cells, you can do it too, but it is much, much, much harder. It is harder. It's not that easy. The advantage of taking adult stem cell, is that right? For example, I play too much soccer, my heart cells are dying, I need a new heart. They take my skin cells and they turn them into my heart cells, and I will not reject them. Is that right? It's from same person, no rejection. But if they take the embryonic stem cells, and I've been drinking too much alcohol, for example, and they want to put these liver cells inside of me, I will reject it because it's not me. They are not my cells. Am I making some sense? That's a disadvantage between embryonic stem cells versus adult stem cells. Embryonic stem cells, you know, you have the rejection problem, but you can easily grow them in the laboratory. Different type of cells. Okay. What does this one say? Embryonic and adult stem cells, I guess I already talked about it. Let's just quickly read it. If I missed anything, he will tell me. Many early embryos contain stem cells capable of giving rise to differentiated embryonic cells of any type. In culture, these embryonic stem cells reproduce indefinitely, as I talked about it. Depending on culture conditions, they can be made to differentiate into a variety of specialized cells. That's what I talked about that. If in a culture you put growth hormone and other hormones, it will differentiate to different type of cells. Adult stem cells can generate multiple, but not all, cell types and are used in the body to replace non-reproductive cells as needed. Here they go. This is, as I talked about it, embryonic stem cells. You put it in a culture and you grow it to different type of cells. Adult stem cells, you take bone marrow cells and you grow it to different type of cells. Same thing as I said on the board up there. Embryonic stem cells, ES, I guess, is called pluripotent. It means multiple different type of cells. So that's what embryonic stem cells are. They are pluripotent, capable of differentiating to many different cell types. The ultimate aim of research with stem cells to supply cells for repairs of damaged or diseased organs, of course. Embryonic stem cells present ethical and political issues as well. Totipotent, these two terms, I'm hoping you would know them for quiz or exam purposes. I bolded them. Totipotent cells are like egg cells that can be developed into entire organisms. Total. Toti comes from total. It means these cells can become entire organisms. Egg, human egg, can become entire human, so they call them totipotent. The cells you get from embryonic stem cells, they are pluripotent. The cells can become a variety of cells, many cells, not the entire organisms. Okay. Gene therapy is alteration of an afflicted individual genes. Yes, gene therapy holds great potential for treating disorders traceable to single defective genes. Vectors are used for delivery of genes into specific types of cells, for example, bone marrow. Gene therapy provokes both technical and ethical questions as well. Here is a clone gene and then those genes can be, by viral capsids, they can be transferred to the bone of the person, and then, of course, be fixed. The other concept, the other idea is genetically modified organisms that you are eating in stores right now. In California, there was a proposal that maybe the food that are genetically modified, they should be labeled. But people didn't vote for it, so you're not knowing what kind of, I'm talking about myself. I'm not knowing what kind of food I'm eating that is genetically modified. Okay, genetically modified, I'm not talking about the animals that 100 years ago, they took a bowl that gave a lot of meat and they gave the sperm of that bowl to many cows that hopefully those cows will give good amount of meat or milk. I'm not talking about that. I'm talking about they remove genes that I showed them to you in the past right here. They remove genes and put them inside of the cells, and now the cow gives more milk. I'm not talking about organic. I'm not talking about organic and non-organic or BTSG, those that hormone that they inject to the animals. No, I'm not talking about any of that stuff. A lot of genetically modified food that we eat, they are organic. A lot of food that we eat that are genetically modified, they are organic. Just like the cotton. You remember I said the cotton, they take a gene and they put it in the cotton plant, so the cotton plant is resistant to pesticide, to pests. So that's a genetically modified organism. Most public concern about possible hazards in centers are genetically modified organisms used as food. Some are concerned about creation of superweeds from transfer of genes from GM, genetically modified crops to their wild relatives. Other worries include the possibility that transgenic proteins produced might cause allergic reaction. So we are eating them and we don't know, they have not been around for a long time, so we don't know their effect. As biotechnology continues to change, so does it use of agriculture, industry, and medicine. National agencies and international organizations strive to get guidelines for safe and ethical practices in the use of biotechnology. Is there any question? Does anybody have any question? What do you guys think?